Fluorescent in situ hybridization, or FISH, is a molecular cytogenetic procedure that uses fluorescently dyed fragments of purified DNA, called probes. These probes are used to bind to a matching set of DNA on a chromosome sample that is being tested. The bound DNA segments can then be found with the aid of a fluorescent microscope. The ability of FISH to find DNA sequences to a high specificity makes the technique valuable in medical and science fields, such as the diagnosis of genetic diseases, gene mapping, and the discovery of aberrant chromosomes, as the ability to pinpoint specific DNA sequences has an enormous number of uses. FISH can be used to compare DNA samples from a child and their parents to determine if the child has any genetic abnormalities, as well as further exploring the nature of those abnormalities if their presence has already been detected. Other diagnostic uses for FISH include the ability to test for abnormalities in sperm, as only a few chromosomes need to be tested for abnormalities. FISH can also be used in cancer diagnosis and analysis to generate a quick result. This is because other cytogenetic procedures require cell division to measure results, requiring a cell culture to be grown, which takes time. FISH tests can be used to exploratorily test the location, amount, and extent of tumours in the body, from tissue samples taken in a relatively short amount of time. Science applications of the FISH procedure include using it to identify new microorganisms, especially pathogens, and comparing the DNA segments of new microorganisms to existing organisms to search for similarities in genetic code. This allows for a more detailed estimation of how a new microorganism might behave. FISH can also be used to determine the population composition of environments with multiple microorganisms, as DNA segments unique to particular microorganisms can be used to show their location.
FISH works through the use of fluorescent DNA probes to target specific genetic loci, resulting in colored signals which can be observed through fluorescence microscopy. The first step of FISH is to heat the DNA of interphase or metaphase cells on a microscopic slide, resulting in the denaturation of the double helix structure of the DNA. The complementary DNA strand is then inserted, which is attached to a fluorescent label. This label allows the targeted gene to be highlighted, allowing for visibility for fluorescence microscopy. The DNA is then cooled in order to hybridize the original DNA strand and the introduced complementary strand with the fluorescent probe. The final step is to view the fluorescent DNA segment under a confocal fluorescence microscope. The physical and biological drivers of fluorescence in situ hybridization are the principles of nucleic acid base pairing, as well as fluorescence microscopy. Nucleic acid base pairing is a crucial element of FISH, allowing a complementary DNA strand to be produced and hybridized, which results in fluorescence. Fluorescence microscopy involves the illumination of a specimen with a specific wavelength of light, which is absorbed by the fluorophores of the sample, allowing these areas of the specimen to be highlighted. Hence, the observation of the genes of focus is dependent on fluorescence microscopy. There are various forms of FISH machinery having different uses. The most common are single or double probe detection kits, as well as quantitative multi-gene FISH kits. Single probe detection kits utilize one distinct fluorochrome to detect an alteration of a genomic fragment specific to a single locus, whereas double probe detection kits utilize two fluorochromes to detect the alteration of a balanced chromosomal translocation. Quantitative multi-gene FISH, known as QM FISH, uses a range of fluorochromes in order to simultaneously detect deletions or amplifications of multiple genes. The display results under a fluorescence microscope would usually show the full set of chromosomes of the specimen, with the fluorescent probes attached to the DNA being clearly visible, displaying the studied genes. The displayed results show the gene for rust resistance being displayed in the genome of wheat plants in metaphase cells. The results shown on this slide display the use of FISH in the field of cancer research, showing the genome of an ovarian cancer patient prior to and post-chemotherapy, displaying the results of the treatment.
Fluorescent in situ hybridization, or FISH, can be found as use of classification of genetic syndrome. FISH works with the FISH probes to provide prognostic information for the clinician. We figured that one of the applications of FISH was used to classify leukemia under donor hierarchical classification. Donor hierarchical classification is used to categorize and identify deletion in chromosome number 17, together with deletion in number chromosome 11, trisomy 12, and deletion in chromosome number 13. These deletions in chromosomes are a common case of chronic lymphocytic leukemia, or CLL, which possibly also lead to DLEU2, or understood as deleted in lymphocytic leukemia 2, which is non-protein coding and occurring microRNA 15A and microRNA 16-1, which is potential cancer gene. The most important reason why FISH is used for research of chronic lymphocytic leukemia is because the FISH panel data can be used whether in early time of leukemia or more chronic progressive status, such as acute leukemia. FISH improves risk stratification in acute leukemia by identifying KMT2A, or known as lysine methyltransferase 2A, which is protein coding gene, abnormality copy, and translocationing. According to the research, we're observing the 1,585 CLL patients in the CRC database over years, which time to first treatment was measured from data of diagnosis to initial time of first treatment. The CRC centers that provided 1,585 CLL patients used standardized FISH panels to make results. This is the distribution of patient deletion in chromosomes by FISH category using donor hierarchical classification, and time to first treatment by FISH category for 1,048 patients who had CLL diagnosed before less than 7 years in the research of the donor fluorescence in situ hybridization prognostic classification of chronic lymphocytic leukemia, how FISH was being applied in research.
I'm going to talk about the second application of FISH, which is the detection part. For the first part, FISH in the cancer detection. This technique is widely applied in the cancer diagnosis, rate facilities, and the identification of the cancer-associated chromosomal abnormalities. For example, in breast cancer diagnosis, the technique is used to detect the HER2 gene abnormalities. Tissues with breast cancer have extra HER2 gene copies, which the technique detects in the cancer tissue obtained through the biopsy. More HER2 receptors in the cell result from more copies of the HER2 gene, and these receptors receive signals that invigorates the growth of the breast cancer cells. For the second part is the detection of infectious diseases. FISH can also be used to detect infectious agents using phylogenetic group-specific 16S rRNA. FISH probes with oligonucleotide sequences complementary to 16S rRNA can be used to study microbial communities in the oral cavities and gastrointestinal flora. In the same way, FISH has been used to detect the pathogens within tissues. Pathogenic microorganisms in blood cultures have been identified using oligonucleotide probes that are genus and species specific. FISH probes complementary to a specific sequence of 16S rRNA, for example, can detect malaria infection in blood samples.